friends. Welcome to our Frankly Speaking About Cancer and Clinical Trials presentation today. I just wanted to introduce myself briefly before I pass the presentation along to Dr. Solwitz in his presentation. It's an honor to be here today. My name is Catherine Scheibel. I am Program Coordinator at Cancer Support Community of Central New Jersey. And for those of you who don't know, we are located in Bedminster, New Jersey. We have the privilege of working in partnership with several medical institutions and organizations like Regional Cancer Care Associates to make educational programs like this happen. So thank you all for being here. I hope that you enjoy the lunch presentation and, of course, the presentation on clinical trials. So for those of you who don't know us, Cancer Support Community is a nonprofit organization. We are a part of a larger international affiliate network where we provide free educational workshops like this, wellness and nutrition classes, ongoing group support and individual counseling to all people who are affected by cancer. It's our goal to address the psychosocial and emotional needs of people who are affected by the cancer diagnosis and to ensure that no one faces cancer alone. So I just wanted to mention some of our resources. Could you just click next on the presentation? You can go to the next one. Okay, next. So some of the resources that are supported by our national headquarters, we do have a helpline to assist you outside of normal work hours if you're ever in need of immediate support, so keeping that number in mind. We also have a program that's called Open to Options. This is a counseling program, and it's designed to prepare you with a list of organized questions, thoughts, concerns that you may have that you can then take to a physician's office and share it with your physician at an appointment that's related to treatment decisions. So we find it helpful to folks who are recently diagnosed, though it's also proven to benefit patients along the continuum of care. So for example, if you are faced with making a treatment-related decision about entering a clinical trial, we do recognize that these are decisions that can be extremely hard to make. So we are here to help you through that and making the decision. And then lastly, Cancer Support Community's landmark educational series are Frankly Speaking About Cancer. These workshops provide evidence-based information on a variety of topics related to the cancer experience. So tonight we will be, or today, excuse me, we'll be addressing issues around clinical trials. Okay? So today we'll listen to Dr. Solwitz's presentation. We do ask that if you have questions, please make note of them. We'll make sure to have a Q&A at the end. I will also follow up with discussing another one of our national programs, Cancer Experience Registry. Okay? There are also evaluations that you've been given, so make note of any concerns and questions that you have along the way, and we truly value all of that information. Okay? So without further ado, Dr. Solwitz.
So when I joined the National Cancer Institute in 1983, 70%, 72% actually of my patients were expected to die of cancer. That number this year will fall to less than 40%, and even that 40% will survive much longer and on average with cancer than they were a decade or several decades ago. One of the big topics, one of the big problems, if you have, for oncologists, for the oncology community, is what we call in our conferences now survivorship. And that is, what in the world do we do with all those survivors? You know, we have people now who are living decades after treatment, and what does that mean as far as monitoring them and long-term side effects? And we have patients that are living for years with treatment, and how do we watch them and take care of them? So what's resulted in this incredible change? You know, why do we have this transformational change in cancer and how we understand cancer? And the answer, obviously, clearly, is research. The answer is science. The answer is the work of the last 50 years. You know, over the last several decades, there have been over 40,000 articles published on cancer research every year. And that number this year may be even higher. We are having more drugs and treatments approved for the treatment of cancer every year than the year before, and this is changing faster than global warming. You know, there's a continued acceleration and transformation. We are beating this disease. And how is this possible? The answer, of course, is cancer research. My goal today is to give you a little understanding about clinical research, cancer research, and how it happens, how it may apply to you, how you can understand what you're hearing and what you're reading. Those who know me, a lot of you, I do know that after 36 years of marriage, I interrupt very well. So if there are questions you have, while I'm speaking, please don't hesitate to raise your hand. I'm happy to address those questions while they're in your mind, and we can talk about that. And we really appreciate, very, very much you coming today, and we'll try to answer all your questions as you go forward. All of you were given, as you came in, a hot-off-the-press cancer research result, a piece of paper when you came in. And just to clear, so that we're right on the edge of everything, this is an announcement from the FDA that came out last evening. Oncologists, get this by email, and this is an email that I received last night. And this is a breakthrough drug that was approved by the FDA yesterday for the treatment of bladder cancer. And to tell you how cutting edge, we actually had a patient in the office yesterday afternoon that we weren't aware this drug was about to be approved and then it just came in the evening and we turned around to call the patient and go, well, let's have you come back. We have a new drug that's available for you, and we didn't know it's about to be approved. The important thing about this piece of paper is that this discusses the research study, the clinical research study that resulted in the FDA approving the drug. It talks about multi-center single-arm study. It talks about the patients that were included, excluded. It talks about overall response rates. It turns about side effects. This is the research study. And one of my hopes today is that we're going to go back to this later, because I'd like you to understand the basic ideas of how it gets to the FDA, approves a new therapy based on this research and clinical trial. How do we get to this point? So this is, of course, a story. And this is a story of many years in getting to this point. The story, I think, appropriately starts in about 1884 in New York City. And before this time, cancer was seen as a disease of rotting failure and of the soul. If you got cancer, you didn't talk about it because it meant you were an evil person and your body was rotting and you deserved to, therefore your fate, you deserved to be yours, you deserved to die. And no one talked about the disease. And if you don't talk about it something and you don't share about something, then no one ever is going to learn about it, and you certainly can't study it. In 1884, Ulysses S. Grant was dying in New York City of mouth cancer, which he got from chewing tobacco and cigars. And one thing about Ulysses S. Grant, whether you think he was a great president, not a great president, great general, not a great general, is he was wildly popular. And he allowed the story of his disease to be covered in the press. And the tabloids, which were just exploding at that time, followed constantly around what was happening to him, including one event in the middle of the night when he started bleeding from his mouth, and the doctors had to get on horseback and rush across the town and rush in and stop the bleeding. And this was all tabulated in detail in the papers. And the story of his disease and his death from cancer became a national sensation. And this changed the conversation. This was a moment in time that people can now start talking about cancer, because if Ulysses S. Grant could talk about cancer, we can talk about cancer. So it started to become part of the conversation, and when then people started to look at it. Now in the 1890s, the first major cancer therapy occurred in this country, mainly around the New York area again, and that is a Dr. Coley noticed that when people got really infected with a pneumonia or a bad urinary infection or some sort of overwhelming infection, that they had high fevers and they were really toxic and really sick, if they happened to survive that infection, some of them, the cancer shrunk a little bit. And he said, oh, we can fix cancer. So he started deliberately infecting people. And this, unfortunately, disgustingly means that if you went to the doctor and you had a tumor growing, he gave you an injection of pus. That doesn't work in my practice, but apparently it did in his. And again, if you happen to survive that, there was a small cancer, your cancer would shrink. Now, this is true. This is so-called Coleys Toxin. It happens due to secondary to release of tumor necrosis factor. Obviously, Dr. Coley had an awful habit of killing many of his patients, and didn't understand all what was going on. But this is some of the first therapy. In the early 1900s, 1905, 1910, people started to play with radiation therapy. And they started to notice that if you took a radioactive substance, you exposed it to a tumor, that some of these tumors shrank. And people were literally taking radioactive salts, radioactive stones, and they were taping them to people and putting them in front of radiation projectors and treating with radiation therapy. Radiation therapy by 1910 was very popular for treatment for cancer. Now, you can imagine the disasters that occurred because of radiation toxicity that occurred in this setting, but this therapy was happening. A critical thing about the next 20 or 30 years was that people just spent a lot of time starting to understand what cancer looked like under the microscope. You have to understand that in 1930 in this country, if you got cancer, there was less than a 50% chance they would get the diagnosis right. They just didn't know what cancer looked like or what it was. But people spent that time period learning what cancer looked like under the microscope, studying the disease, and that was a basis of starting to build just some basic understanding, even though the vast majority of patients continue to do very badly. Now, there's a side note, and this is important for cancer research and all medical research, but even though it wasn't a cancer event, that started happening in 1932. And in what has since been called the Tuskegee experiments, they started doing a series of tests on sharecroppers, African-American sharecroppers in the Deep South, where they started experimenting with syphilis. And this was a horrible, in retrospect, series of experiments where they took men who were known to have syphilis, or men that didn't know to have syphilis but were at high risk, and their families, and they started tracking them and following them. Now, what they didn't tell them is that early in the experiment, they lost the funding for the treatment part of the study. So they started just following these men and never told them, by the way, they had syphilis. They told them they had bad blood. And they started following them for decades and decades and decades just to see what would happen to the syphilis. Now, this was particularly tragedy because by 1947, we knew that penicillin cured the vast majority of syphilis, a single injection, and these men could be fixed. But none of these men received treatment. And they basically tracked them over time to see what would happen to them with untreated syphilis. This was eventually unearthed in a 1972 newspaper article and became a disaster, for the Public Health Service, and these people, there was lawsuits and everything. But a vast majority of these patients had gone on to die of syphilis without ever being treated. This is critical, the Tuskegee story, because it is the core basis for much of the ethics in how medical research, and particularly cancer research, is carried out. It is the basics for consent. It is the basis for information. It is the basis for the entire structure around it. People talk about, I'm concerned I don't want to get involved in a clinical trial because I don't want to be a guinea pig. I don't want to be a placebo if there's another treatment. Well, the Tuskegee experiment and all the ethics studies and everything that followed it, is the core basis by which we don't do studies like that. It was a critical learning experiment. Now, in 1945, just near the end of the Second World War, an American ship was unloading mustard gas in Italy. Yeah, we actually did use some mustard gas in the Second World War, although that's something has been suppressed. And the mustard gas container as the stevedores were unloading off the ship, released, unfortunately, and the canister of mustard gas fell onto the wharf and exploded. And a number of different stevedores and other people working around the can, people on the ship, were gassed with mustard gas. And they were immediately rushed to hospitals in Rome, and many of them died, but many of them were very sick, and they survived. What was important is that, of course, we all know that millions of men and women were gassed in the First World War. But in the First World War, we didn't understand as much about the biology. I talked about that time frame from 1910, 1940, when people were doing a lot of basic studying in biology. And so what they were that basic time frame. So now we had this science. So they looked at what was happening to these men and women in Italy that had been gassed, and they noticed something about them that would never have been recognized during the First World War, but now is recognized. And they noticed that all their bone marrows were wiped out. that they were all had severe depression of their red counts and their white counts, what you guys kind of say, oh, that sounds like chemotherapy, and you're right. And so the doctors said at the time, this is a substance, mustard gas, that maybe we can use to treat diseases, like maybe cancer, that may be based on blood cells. So this information was relayed to some researchers in Yale, and in 1947, a modified version of this substance, known as mustargin, was given to a series of patients with Hodgkin's disease. And lo and behold, the disease melted away. Now that is a mind-blowing observation at the time. I mean, this is, no one had seen anything like that ever happen with cancer. But don't forget, it was happening at the same time we had started to have the first antibiotics, sulfa, penicillin, the antibiotics that formed. And suddenly they said, whoa, cancer is going to be like pneumonia, or it's going to be like syphilis, et cetera. We're going to be able to just give it a drug and get rid of it. So people had the idea suddenly that you could use drugs to kill cancer. And people started to develop the idea to go looking for this. Well, how are we going to do this? How are we going to go find these different substances that are out there? We can't just wait for accidents to happen like in Italy. So in 1952, the National Cancer Institute was founded. And the National Cancer Institute, for the first time, brought organization to this structure and started to direct the focus of cancer research. And the NCI did a number of major changes in how we were looking at cancer. First of all, it was the first war on cancer, if you will, the first start of the focus. So that was the first thing. It started to bring it forward. Secondly, the NCI, from the start, started to supply money for research and started to build research, so that people in parts of the country and everything could do that. And then the reverse, the NCI also started to coordinate the conversation. So that if somebody was doing research in San Francisco or somebody was doing research, in New Jersey, people understood what was happening and they started to share that research. The NCI started to develop cooperative cancer studies, so that if you wanted to do a study, but it required 1,000 breast cancer patients, that would be really hard to do in just, say, New Brunswick. But if you brought together 10 hospitals around the country, multiple groups together, then you could get a study done. You could get a study done on their way. The NCI started to fund and organize that. The NCI started to push out ethics and standards for research. And then importantly, the NCI started to develop a mechanism, a system for finding these drugs and then what you did with it. Now, what the NCI did then, and it ran for the next 25, almost 30 years, is they started to do something that sounds very crude, because it was, but it was in the end relatively effective in finding cancer agents. And they developed a screening program. The screening program was pretty simple. You took mice that had tumors, and then you injected them with anything you could find. So if you can think of a chemical in your world, they injected them with these chemicals. Any chemical in any bottle sitting anywhere, any cleaning detergent, any paint, any food, any plant, if they could find a chemical, they injected it. And you're talking about taking like 300 mice with cancer and injecting them all with the substance to see if one or two of them were, one or two of them. The NCI screening program screened over 1,000 substances a year at 300 to 400 mice per substance for the next 25, 30 years. And the NCI screening program, which was national, which is a massive project to do all these, find the chemicals, purify the chemicals, find all the animals, ship all the animals, this big industry that was built around this. And the NCI program, on an average, found one substance a year. On average, they found one substance a year. It turned out about 1 in 750 substances that got screened in that range, turned out to have some activity in cancer. Now, some of them, of course, even though you find it, you go, great, cyanide works great in killing cancer, which is a true substance. Not a very effective agent for human beings to treat with cancer. So a lot of the stuff, because you found it, didn't mean it was usable. But over time, on the average, about one substance that would be usable in human beings was found. And most of the anti-cancer agents, the classical chemotherapy agents that were developed from the 1950s into the middle of the 1980s, were found by this program, just kind of grinding through and grinding through and grinding through and grinding through, to find individual agents. Now, let's see. What's your next slide? I'm trying to figure where we are. Okay, so I guess we'll stop. Yeah, that's okay. Fine. So so that program ran for for that period of time. Now, starting in about the early 90s, you had started to have a change in how we find cancer agents and cancer drugs. And what started to change is that we understood the human body better, we understood the immune system a little bit better, and we understood cancer a little bit better. And people started in the 1990s, it's not long ago, to specifically try to find drugs that specifically treated something in the cancer. So for example, you could find that the cancers had a particular metabolic pathway that was particularly more active. And then you'd say, okay, I want a specific substance that shuts down that specific metabolic pathway. So in the 1990s, you started to find drugs that people went deliberately looking for a specific pathway. And we've seen now the transformation since the 90s to now to that method of looking for anti-cancer substances. Now, basically all cancer agents that are now in development, that are now in clinical trials, that are now being approved, are found deliberately because we understand much more about cancer, and we understand much more cancer biology and the immune system. So specifically now, you're saying this gene causes this cancer to grow. We need a substance that shuts down this gene. Therefore, we're going to look for a substance in the gene. Very specific. The cancer has this revved up metabolic pathway. There is this flaw in the immune system that does not allow the immune system to track the cancer. We're going to fix it specifically. So basically, everything now that is being developed is found in a deliberately measured fashion. And more, those things are being found by computer modeling, and clearly the next decade we'll see that you don't even necessarily, you'll model the cancer in a computer, the computer will identify a pathway, you'll look for a substance, and the computer will help you design the substance, and you'll go out and make the substance. And that is clearly what we're having here. But so far, all we've gotten to on the story is you have a substance in your hand. How in the world do you figure out what you want to do, how you do with people? And this is the concept of clinical trials. This is the concept of how do you actually figure out if it works in people and how do you get it there. Now, every substance that has come for clinical research first goes through the basic screening process of does it appear to be active against cancer at in the cell culture level. I mean, literally growing cancer in the petri dish, and you put a drop of the substance in, does that work? Now, just because something works in the test tube, in the petri dish, doesn't mean it works in the human being. I believe in human beings enough that I think we're a little more complicated than the average petri dish. And that's true very much so because when you talk about cancer, you're talking about a disease that affects biology, it affects physiology, it affects the immune system, it affects genetics, it affects secondary systems. And in the human being, cancers fight back. I mean, it's much more complicated than a petri dish that you could use a hammer against. So the first test, however, every substance starts is basically at that cell level. Then they'll start at the lower animal level, and unfortunately, mice continue to be our victims in this. Nowadays you have what are known as nude mice. A nude mice has a very limited immune system itself, so you can grow a human cancer in a mouse. And then you can use a therapy in the mouse and stuff and see if it works, and you can do that. And then you're talking about a lot of use of dogs and some use of monkeys and higher animals and stuff, and you're learning, does it work? And you're trying to get a handle on what are the side effects. Again, human beings are more complicated than some other animals and stuff, so you try to get a handle on the side effects of what the side effects might be. And so by the time a substance is ready to even think about getting into a human being, it's had a whole level of screening, right up, which gives you a feeling of what are the side effects, is it likely to work, and a basic concept of what is the dose. And that's what gets you up to the basic level of starting to move into human beings. And this whole area has been what's called translational research, or bench to bedside. And that is, you have a substance and you walk it through the steps going from the bench or through eventually to the bedside to the human being, and then in a true translational research where you take the observations from the bedside and you bring it back to the bench. Now let's see, what's the next slide? There's one that goes next, nope, good, next. I say we're almost there. Good. All right. So the NCI came up with this model a long time ago. The model still holds. it's getting more, computers make it more and more easy to do in a lesser amount of time and safely, because you can look at a lot more data, and analyze it. But you still have the problem that still the first patients have to get, have to be the first people to get a drug. And the first step is what is known as a phase 1 trial. Now, a phase 1 trial, the goal of which is to find out, truthfully, is what it can be given safely to human beings, what is the side effects, and what is the true dose. Now, you notice I didn't say anything about treatment in that sentence, because that's not really the goal of a phase 1 trial. Now, obviously, these are substances you have reason to believe have anti-cancer activity, and they may help in human beings, but you don't much of the time know if they're going to help, how they're going to help, and you don't know even what cancers they are likely to help. And a phase 1 trial is a toxicity trial. And it's usually a small number of patients, several dozen, and it starts at the lowest possible dose that makes sense based on animal studies, and then basically, usually in phase trials, you're just increasing the dose till you get to the point of finding what the side effects are, and what people can and can't tolerate. I think there's one more slide on that. So, as it says here, they're not actually supposed to test how well a new drug combination of drugs works. The phase 1 trial is a toxicity trial. So why would someone get involved in a phase 1 trial? And the answer is a patient wants to volunteer their life and their body for the opportunity to perhaps help others, and they have the possibility, perhaps, that it may help them, as long as they understand that is not the goal. Now, I want to be clear. People that enter a phase 1 trial are not like Tuskegee. Phase 1 trial, the researchers sit down with you and go, I just want you to be clear what you're doing here. So this is, we're studying the drug itself. We're going to watch what happens to you. We're going to take really good care of you. You're going to get the right x-rays, the right tests, et cetera. Keep it very clear. You're not a guinea pig in the sense that we don't care about you. But clearly, you have to understand what the goal is of a phase 1 trial. And there's no magical way to get by this. Now, like I said, computer modeling has made this better and better. The better use of animal models gets this better and better. The likelihood you're going to give someone something that is just a disaster in human beings immediately has gotten smaller and smaller. But a phase 1 trial is just a side effect trial. The reality exists historically. Phase 1 trials have been of benefit to the patients in less than 10% of the time. It's a small number, because maybe it's going to work in breast cancer, but the person you're testing it on, it's a lung cancer patient. Or maybe it's going to work in lymphoma, but only combined with another drug. So, because this is all much more complicated. But phase 1 trials are critical. Hundreds of thousands of men and women over the past, since the NCI started this, 1950s, have been involved and volunteered for phase 1. No treatment that any of us has received for cancer has not been through a phase 1 trial. It is the foundation, of the start, of cancer care. Now, if a drug gets through phase 1 and people can tolerate it, and it looks like it has some benefit, because you do watch for benefit, you try to get clues. You try to say, well, look at breast cancer, work to lymphoma, whatever, et cetera. You try to get clues. Then it will go on to what's called a phase 2 trial. All right, now a phase 2 trial is an efficacy trial. It is a benefit trial. The goal of a phase 2 trial is actually to try and help patients. But it is given to focus on a particular disease. So you take the information from the phase 1, you take the information from all the biology, you take information from what you understand about the treatment, and you will give it, it will be given for a specific cancer in specific patients to a specific dose. Phase 2 trials can be quite large. They may include hundreds of patients. And the goal of a phase 2 trial is actually to get benefit and see how well a drug works. And fortunately, thank goodness, we've gotten better and better at understanding about when phase 2 drugs are going to work and not work. We have less and less chance by the time it gets to phase 2 that it's not that you're going to be wasting a patient's time. Now, a phase 2 drug might be using a particular new chemotherapy agent to treat breast cancer, take bladder cancer, treat lymphoma. It could even be a combination of a couple things given together. And the goal is to try to, help a patient. Phase 2 trials are going to be much larger. Now, this is the situation people worry that I'm going to get on, because a phase 2 trial, you have to compare it to something, right? I mean, you have to know if it works better than what you're already doing. And patients worry, and they get involved in this kind of a study, that they're going to be getting, that it's going to be, that they might not get the study drug, they might get a sugar pill, a true placebo. A phase 2 trial is usually, not always, a randomized trial, which is to say, usually there are at least 2 arms. One gets the new substance, one doesn't get the new substance. But the arm that doesn't get the new substance at least gets the best standard care. Nobody in 2016 would give one group a new experimental substance, but give the other group nothing, if there was something else possible available. So these studies are randomized against the best possible alternative. Now, sometimes, if you look at the study in front of you, which we'll come back to, that's a phase 2 trial, but there's no randomization. And that is, the reality is because there's no good alternative for that situation that's described in the study in front of you. So everyone on the study got the treatment. We don't, that would be like Tuskegee again, randomizing people doing nothing. So a phase 2 trial includes, if it's a randomized trial and has it has 2 arms, or maybe more than 2 arms, it includes at least the best alternative. And again, the goal is to watch very closely, to give the best treatment. And actually, people in clinical trials frequently get better treatment, or in the sense of how often they get tested and how closely they get watched than on standard therapy, just because they're watching the studies so closely. In fact, people in studies will sometimes complain they're getting too many tests and studies because they want to know every side effect. We want to know if it's the benefit. They want to know when there's benefit. So people will watch very, very closely on that. Now, sometimes in a phase 2, many times in a phase 2 study, if it turns out that the study drug is causing benefit and it's detected during the study early on, that the study, because these studies are all very closely monitored, what is happening to all the patients. And it turns out early on they detect that there is a benefit from the study arm, which is better than the standard arm, than the standard therapy, then the monitors of the study will close the study. And this simple say it does not become unethical at this point to continue the control arm and not give people the new study drug, because we've proven it's a better treatment. And the monitors will close the study early. Or what will happen with many phase 2 studies, if that doesn't happen, is that built into the study will be the possibility of being rescued or crossing over to the other arm. And that is, you start on the standard therapy, and then there's there's an experimental arm, and if you're going along and you start to fail on the standard, you can cross over the experimental. And the reverse may also be true, that if you fail, you can cross over. All these things are built into the study from the start. The monitoring, the timing, the length of the study, the number of people on the study, the doses of the drugs. This is all completely built in. And patients consenting for these studies are informed about this. Now, if you go on a study, they give you a form which seems to go on forever. And the reason is because this is what they're trying to make you understand what is going on. And it is the obligation of the researchers on the trial to explain to you carefully how the study works and stuff. Is there a crossover? How long can you continue on the drug if it turns out the drug works but the study finishes? How are you going to get the drug at that point, but if it's not FDA approved yet, which most studies allow for? All these things are available up front. And then what happens if this drug doesn't work at that point? Where do you fall back to? Do you go back to standard therapy? What are your alternatives at that point? And many these studies are done on a continuous basis, and phase 2 studies are really critical. And much of the time when people are talking about new studies and new drugs, you're talking about at phase 2. Next. So phase 3 studies are where stuff gets much more, more complicated. And you're starting to combine therapies that work in new and complex ways. So say I want to know the treatment for inflammatory breast cancer. Now, inflammatory breast cancer is a particularly aggressive and lethal cancer, which includes, for every patient, the combination of chemotherapy, radiation, surgery, and often hormone therapy. And I got chemotherapy that I know works, and I know what the surgery is, and I know what the radiation is, and I know what the hormone therapy is. How in the world do I figure out what order to give them in? Do I do the radiation followed by the chemo, followed by the surgery, by the radiation, et cetera? What order? What order do I do them in? How do I organize that? And the answer is a phase 3 study. And a phase 3 study starts to make combinations of, okay, so let's take the inflammatory breast cancer example. I'll do the chemotherapy first, but then I'll go directly to surgery, and then I'll do radiation. Or I'll do the chemotherapy first, and then I'll do radiation, and then I'll go to surgery. Or I'll do the chemotherapy, I'll go to radiation, but maybe I won't do surgery on everybody because I'm not sure they need it, so I'll randomize people to get radiation, no radiation. And you can work out the math of how complicated these kind of studies are. A phase 3 study is when these things start to get complicated. As you can imagine, phase 3 studies are immense. I mean, phase 3 studies are complicated studies that require multiple institutions, in multiple places around the country or even around the world, because they may require 3,000, 4,000 patients just to answer some of the simple questions. Don't forget, many of the times when we make breakthroughs, we're talking about a several percentage breakthrough. Now, several percentage, when we're talking about thousands of lives, is important, but detecting it during the course of a study, can be extraordinarily difficult. So phase 3 studies are what is when you're really out there in the real world and you're building the basis, of long-term care. And a lot of the stuff you see published, years later, people are complicated long-term phase 3 studies. Now, there's also the concept of a phase 4 study. Phase 4 studies, I think we have up here. So phase 4 studies, is got like long-term monitoring in society sort of studies. And when we talk about survivorship, a lot of that is phase 4 studies. So someone is treated, for leukemia, acute leukemia, and they get a bone marrow transplant. So we watch them for we're watching from now 5 and 10 and 15 and 20 years. What were the effect of the treatments they had? And what do we talk about? Do they have long-term heart problems? Do they have long-term problems with their bone marrow, et cetera? And phase 4 studies, a lot of this long-term monitoring, ongoing studies. Phase 4 studies usually may involve universities, big systems that's kind of at that level where they're putting all their patients together in a way that the patients aren't directly involved. Just looking at pure data, it's kind of that level, of what is happening. So, and that's basically brings us to where we are as far as this structure of clinical research and the structure of these studies. Now, why would a patient not be involved in a research study? Why, in a situation are you not on an experimental trial? And there are several answers to that question. I mean, the reality is only 4% of cancer patients in the United States get involved in a research study. You're still talking about large numbers of people because, tragically, as you're aware, we have 750,000 new cases of cancer in this country every year. And that number, unfortunately, as the population ages, is larger, even though we have less cancer per patient, but the population's aging. So why would you not get involved? And the first and most clear answer is that the standard proven therapy is shown to work. And the data says that the potential benefit of an experimental trial is less than the standard therapy that's involved. So that would be the first reason. Another reason would be that your particular situation does not allow one to get to be involved in a research trial. Which is to say that if the research trial required you never have had a previous cancer, but you'd had some other previous cancer, that may bring you off the trial, or have other active medical problems which interfere with getting involved in a trial, or your general functional status and your strength didn't let you get involved in a trial. But beyond those fairly limited reasons, well, that's not too limited. Having a treatment that's already working is a pretty good reason. But beyond those reasons, it's reasonable to think about looking at experimental trials at each point in your cancer care. And hopefully your doctor is. Now, the reality is, just like that email, that I gave you from last night, in 2016, and everybody's kind of connected, and hopefully your doctors have easy access and are being informed of constantly clinical trials that are available for your individual disease, and the Versus knows how to find them easily. So hopefully your doctors think about this at each stage in your treatment. But at each stage in your treatment, where you're looking at a change in your status, something new has happened, at that moment it's worth saying to yourself and saying to your doctor, is there any clinical trial available at this time, that I might want to get involved in? Now, maybe there is not. Maybe it's just physically not convenient. But the question is never a bad question. And this is not hard to find out. It is incredibly easy because the National Cancer Institute has made it incredibly simple and straightforward to do. If you go on the NCI's database under research studies, and there is a physician access and there's a patient access, intake in there. There is basically a tabulation of essentially every major research study going on in the country and most minor ones. And it breaks down easily into disease and the geography, et cetera, and it gives you a detailed contact list. So your physician knows this. The physician is hopefully there on a frequent basis as well as other sources. But it's also a place for you easily to look and see, is there something available to me that I should look at? So whenever there's a moment in time that there's a significant change in your therapy, you should probably be thinking about it at that time. Now, there may not be something available, or it may be that standard therapy is the right thing for you at the time, but it's a reasonable question to ask. Now, don't forget, studies don't only involve the treatment of the cancer itself. Studies sometimes involve, should you have more x-rays or less x-rays, the other use of vaccines, other drugs to treat side effects. It's not just you're treating the cancer therapy itself. But you should, at that point, having a conversation, with your physician. Now, whenever I send people on the internet to go looking for research, I always like to kind of try to warn people. So the internet, from my standpoint, is great for bringing up the conversation, but does have the habit of scaring the bejeebies out of many of us, because unfortunately the internet's got everything, and it's, and some of this is at a level of sophistication that's kind of hard for people to understand how it relates to them. So anything you read there, I always recommend, read with a gigantic grain of salt, but then print it out and bring it to your physician. I'm always actually very happy when people come in with an inch thick of, internet printouts, because I know we're going to have a good conversation, and I know people are going to understand, a little bit better what's going on. So whatever you look at there, obviously, read, with a grain of salt. But that's a good time to have a conversation and go back. So, what's available as far as research. And a lot of times research is being done right in your own community. A lot of time it does not require a distant trip. Sometimes, if there's something special, it may be worth getting, in a car, train, plane. But more often the time, this stuff is right here, and you have an opportunity to talk to your physician and stuff and deal, and talk about what choices are available, as you move forward. One important concept that is important to bring up at this point, is the concept of what is called, people call complementary or alternative medicine. And because it has everything to do with research. There's a $60 billion a year alternative and complementary medicine, culture in our society. Many of our patients, do all sorts of complementary alternative treatments. And people, say, well, what's the difference between an alternative medicine, and a conventional medicine? And the conventional term for that is allopathic, conventional medicine. And the answer is frequently just the research I've described. An alternative practitioner is often presenting information to you that doesn't have this robust, this depth of research behind it. Hopefully your standard physician, if asked, could show you each of these steps that I've described and how this is produced. And alternative medicine doesn't mean that it doesn't work. It doesn't mean it may not be of benefit. It simply means it hasn't been tested. It's been tested, then we would have been through this conversation and we wouldn't, it's not alternative anymore. We understand, whether or not it, works or not. So, Laetrile, amygdalin, which is, from apricot pits, became very popular in about 1930. That's when it was first developed, and had a gigantic following in this country, through, the 40s, 50s, 60s, et cetera. It didn't make any sense to people, in standard cancer research that it should have a benefit. It has a trace amount of arsenic in it, and people say, well, that has cancer benefit, but it had a gigantic amount of following. So eventually the NCI got, was getting so barraged that it decided to test it. So it did research on it and actually did some of the, it actually tried to do a phase 1, had some trouble finding toxicity levels, et cetera, but then it did research on it, basically did a phase 2, and it found no benefit. And so amygdalin, laetrile, shouldn't be seen as an alternative therapy anymore because it's like that other thousands and thousands of substances that have been tested and didn't succeed through, through this therapy. Alternative, that's basically the difference. I mean, it's not fancy that, that alternative medicine may not be correct. It simply hasn't been tested. And that's an important thing when you're judging treatment you're getting. Complementary medicine, I put separately. First of all, complementary medicine is frequently much less intrusive, than chemicals. If you're talking about, acupuncture, you're talking about Reiki, you're talking about, et cetera, and that much more supportive. But alternative medicine, I mean, and that's the difference when you start, talking about this kind of research. So you have in front of you the study from last night. So take out your homework. So this was a study. So the FDA within the yesterday announced the approval of the drug Tensotriq, which none of us have used yet in treatment, for the treatment of bladder cancer, which is spread. We have some chemotherapy agents for bladder cancer, which is spread. They can have benefit for some significant period of time. They're not for cancer that is spread, viewed at this time as curative, although some patients do very well. So you're looking at here is a phase 2 study. All right. So if you look at the third paragraph, what's the first thing you see? Multi-center. What was I saying? You can't do these things in a single study. You can't get enough patients together in a short period of time. So the FDA, the FDA doesn't do studies. The NCI organized a multi-center study. It's a single-arm study. And we talked about that. So they decided that there's no good alternative for metastatic bladder cancer for people that have already had chemotherapy. So this study does not have a second arm. There's no randomization. There's no... there's nothing to balance it. So every single patient on the study got treated. And that's what that term single arm means. How many patients? It took them 310 patients to answer the study. The next thing it says, it defines who the patients are. Studies are very clear. So it says patients with disease progression, the cancer increased, following a platinum-containing regimen. Platinum is our most active drug against bladder cancer. So that's cisplatin, that's carboplatin, et cetera. All right, now, the next study is important because who can't go on this study? It excluded patients with an autoimmune disease or systemic immunosuppressive medicine. Now, why did they do that? Why did they walk in the door and say, well, if you've had an autoimmune disease, you've had lupus, rheumatoid arthritis, psoriasis, bad psoriasis, you can't go on this study. Why? Because the phase 1 studies told them that those people got sick. So this is a PD-1 inhibitor. This drug is so bloody cool that it's just, this is PD-1 inhibitors are these new immune class of drugs that is changing all of oncology. So you remember Carter stated all the cancer that spread to his brain? PD-1 inhibitor. Melanoma that spread through the body, people now getting 50% major responses, PD-1 inhibitor. Metastatic renal cancer, PD-1 inhibitor. Lung cancer combined with PD-1 inhibitor and a checkpoint inhibitor. What do these drugs do? Why doesn't your immune system cure the cancer? If I transplanted a kidney into any one of you, your immune system would destroy the cancer. Why doesn't your immune system destroy the cancer? Because it doesn't see it. It comes from your own cells, and it thinks it's a normal part of breast or lung or pancreas, wherever it came through. Your immune system doesn't see it. And what stops that? Something called the checkpoint. The immune system is turned off. It's designed not to kill self. It has a checkpoint. What is a PD-1 inhibitor? It's a checkpoint inhibitor. It turns off the checkpoint. We didn't know this 30 years ago when people started looking for cancer. We didn't understand the immune system at this level. This is incredibly bloody cool. So this is a checkpoint inhibitor. But when they tried to use it in the phase 1 studies, they find that people that already had too much immune activity, lupus, rheumatoid arthritis, psoriatic arthritis, autoimmune diseases, that if they gave a checkpoint inhibitor to these people, they got really, really sick. Their lupus flared, their rheumatoid arthritis exploded. So if walking in this study now, they said, we're sorry, but if you've had an autoimmune disease, you cannot be included in this study. Then the next line, the next time shows you one of the pain in the butts for these kind of studies. Tumor specimens required in all patients. Any of you that ever been involved in a study can't believe how many questions they ask, how many tests they want, how they want to have every paperwork, how long it takes to get organized, because they have to have absolute, complete information. And so where it says tumor specimens were required, I guarantee you that wasn't just required. It had to be a certain amount of tumor. It had to be a certain time period after the biopsy. There's a certain amount of tissue, certain tests, all sorts of restrictions. It just shows the restrictions. Okay, the next thing is really important. All patients received an IV infusion of itolizumab, 12 milligrams every 3 weeks. No placebo here, guys. Every patient was treated. We want to find out if it works, etc. And then the last sentence means major efficacy outcome measured including. What that says is before they started this, they said, this is what we're looking for. Before we start this, this is what we're going to monitor. This is what we're going to watch. The next paragraph is the description of the patients as they walked in the door. 78% of patients had cancer in their lungs or liver is what that means, basically. All right. The next sentence is what they measured. They confirmed ORR, overall response rate, by independent review. Now that's important. Independent review. So if I'm doing this study and you're my patients, I don't get to look at the results. Because why? Because I'm biased. First of all, I got a $2 million NIH grant that's dependent on my career, is dependent on this, and frankly, I know you, and I'm committed to you, and I love you. Therefore, I don't get to look at the results. Independent review. So they take the x-rays, they take the blood tests, they take all the specimens, and somebody else that doesn't even see you, and I don't even meet, gets to look at the information. All right? That's what that word's independent review. It is critically important. So most of it probably gets shipped to the NIH. They ship the x-rays, the slides, the blood tests, and they say, and that's the person that's looking at this. I don't get to, as you're treating, do. Now, it said, no, you get a 14.8% response rate. Now, I think all you're going, 14.8% response, I mean, what a terrible number. I mean, only 15% of patients responded to the drug. Okay, who are these patients? These are heavily pre-treated patients who have had significant cancer that is spread, and an overall response rate means a greater than 50% reduction in cancer. All right? So this is a significant major response in a subgroup of patients who were in deep trouble. It means that if we treat people, it doesn't count people that had the stabilization of disease, that the cancer just stopped growing. It doesn't amount to people that had a 49% reduction in cancer. The cancer, 48%, it means 50% more. And then it doesn't state how many of these, it doesn't say how many completely vanished. It's just they've lumped everything together. This would be considered, and this is also a single agent study. They didn't combine it with anything else. They didn't combine it with radiation surgery or anything else. This is one. This would, in the cancer research world, this is a significant major response. And one of the major response. So this is a significant, clear benefit. And then they notice in the next line that of the 46 patients that responded, 37% of the response payer had a greater than 6 to 12 months response rate. So this treatment continues to work for a long period of time. Next to the last paragraph is really critically important too, and that is they continue to watch and collect data about side effects. Again, people in studies are not guinea pigs. We want to know what's going on. We want to eliminate side effects going on. The phase 3 studies that will be built around this will involve combining with other drugs, but it will also involve now how do we get the side effects better? What do we do? How do we prevent side effects? Blood count side effects, nausea side effects, fatigue side effects, et cetera. So side effects are really incredibly important. The FDA takes this information, et cetera, and with it approved a new drug. And it's a new drug that wasn't available yesterday morning. And this is because of these 300 patients that get involved in a phase 2 study, and now all of our lives and the some 25,000 patients with metastatic bladder cancer right now in this country, their lives have significantly changed. Plus, we learn more about a drug, that may change lives as we go forward. Cancer research is critical, to our future. We have new drugs being approved all the time, new therapies being approved all the time. Our ability to understand this disease gets better and better. This is a disease we're clearly beating. This is a disease that we will see gradually vanish in short-term years to come. And this is why. And thank you very much. Huh?
I got a lot of questions for you.
So we'll start. Questions. Okay, so the question is...
and all of that prior to the surgery.
Right. So the question is, patient had lung cancer. The lung cancer appears to be localized by PET scan and other tests that were done, and the tumor is being removed by surgery. The question was asked about studying the patient's blood tests as well as the tumor itself prior to surgery and then presumably after surgery. And this is another type of a study. This is another type of research, trying to understand the natural biology of the cancer, the changes in the immune system that occur when the cancer is removed, et cetera. And this is the kind of study that people often get involved in, even though the therapy is standard, a surgical resection of the tumor, but you're still the opportunity to do more research and learn around. And this is a very common therapy. If you come into Robert Wood Johnson emergency room today or tonight, et cetera, you will be randomly asked whether or not you can have your blood tested for hepatitis C. We're just interested in how much hepatitis C there's out there in the population. And we're just checking randomly right now in the New Jersey population how much hepatitis C is endemic, et cetera. Now, could that help the patient? It might, because we might say, by the way, you hepatitis C, you didn't. But the real goal is to understand the population. So this, yes, and it helps about biology, population. For the individual patient, it's the opportunity to be involved. Perhaps eventually there may be something to help the individual patient, but it's about helping us all.
But what we find the difficult part now, okay, the surgery was only 5 weeks ago. Okay, and where do you go from here? Okay, you talk to your surgeon, your doctor. Does the Cancer Institute play a role in this, of seeing the doctors there afterward to look at pathology or stuff like that, or you go out of the cancer center to...
So who's going to follow you over time? You asked the question, who...
I don't mean the study.
Just off the study.
Right. Forget the study at this point.
So I personally believe that every cancer patient deserves a medical oncologist in their life. That's not just because I'm lonely. And so I would say that if you've had cancer surgery for a real cancer like a lung cancer, that was reasonably monitored by the surgeon, it's probably a good idea to have a medical oncologist involved.
Right, and that's not what I meant. Is that something that you can do at the Cancer Institute, or do you go out of the cancer?
Either one. Either way. Either one. Whatever's you're most comfortable with in building a relationship. There are good medical oncologists in the community that can definitely follow someone over time, and the Cancer Institute also have some really gifted people also.
It just seems a little confusing there, which path to follow.
So then I would suggest that somebody's not. Then if you're confused. So whenever you're on the oncology path, which is a long, complicated path, even for the simplest disease, you should have a champion. You should have someone that you can turn to and ask every question to. And if you don't feel you have that person, you should go find them. And sometimes that just means walking into the original treating physician, medical oncologist, surgical oncologist, whatever it is, and saying, listen, I need you to explain what's going on. I need you to walk it through. Frequently maybe that's calling and saying, listen, I need a little more time. Call ahead and make an appointment. I need a little more time today, and let's organize this. And that is something, frankly, a medical oncologist should be doing to move you through the steps as you go through. So the question was asked, I don't know if people saw 60 Minutes Sunday night. They had the thing on the polio virus. And actually, there are two things. You talk about that, and you talk about fast tracking, which I want to comment on. So the question he asked, has that kind of technique been used, particularly in leukemic in diseases, et cetera? So one of the challenges that cancer therapy has is how do you get the bloody drug into the cell? The solution to that has been to give a whole bunch by IV and hope you soak the person so much that it penetrated the cell, and that's been the solution historically, which works to some extent, but has a lot of side effects and is complicated. So one of the things people have tried to find is, let's use the term magic bullets, and that is a way to take a cancer substance and have it target and get into the cells. And so one of the questions people asked a long time ago is, well, what gets into the cells? Well, what gets into the cell is viruses. I mean, every virus that known to man, the way they work is they penetrate cells. And so the idea a long time ago was to connect various sorts of anti-cancer agents to viruses or virus structure to virus shells. And the poliovirus has been one of the vectors that people have used. People have used a number of other different viruses. You mentioned HIV, MIC has been used, a number of other viruses have been used in order to try to penetrate, penetrate the cells. And this is still an experimental idea. It's a still very complicated and expensive idea as far as treating an individual single patient. But it is an area, and people have played with it a little bit in leukemic. I'm not sure with a lot of success yet, but that's the idea of using this, to do that.
So it's the virus. The virus is not.
The virus is the virus is the transport. The virus is the suitcase. You connect it to a gene that can downregulate cancer cells, or you connect it to a chemotherapy agent or an immune agent, and the idea is to get it in. And then you can kill the cancer internally. Now, the other thing you mentioned, which is right, or that's the idea.
We're doing the dosages also that they're shocking in the first phase, that they actually found it through this.
Right, you don't need much dose because it goes right into the cancer.
It's unheard of.
Right.
And he said they were going for the doses, the higher, they found out that was actually worse. Now they're going like 20%.
Right, and the idea of you have a targeted therapy, it can be used in very low doses. And that's the same thing as using monoclonal antibodies and other things to target. Now, the other thing you mentioned, by the way, was FDA fast tracking. And the FDA has heard in the last 10 years that their process is too slow and has developed a whole bunch of mechanisms for moving therapies faster through the chain, which cuts down costs and particularly time. I have a friend of mine who's a senior executive at Bristol Myers Squibb, and he said that suddenly they have a massive problem is that they can't keep up with the FDA. Which they said is like, in 20 years, he's never seen anything like it, that they send something to the FDA expecting an answer back, in 5 months, and then 2 weeks later, they're going, okay, where's the next piece? And actually, the drug companies are now, for the first time ever, on the other end of that conversation, trying to keep up the FDA needs. Now, there's a line which people are concerned the FDA not cross, which is, how far do you go back? We described, so the study you guys just looked at, wouldn't have been approved 10 years ago. And it wouldn't have been approved 10 years ago because the FDA wouldn't have accepted a single study for approval of a drug, no matter how good it was. The FDA would said, that's good, but do it at least twice or maybe 3 times. So that's an example of FDA fast-tracking what you saw. The concern that people have, and it's just one of these, and people are very aware of it, and they're doing everything to computer model it and work around and listen to it, is the possibility that you could fast-track something comes through that it actually has some significant major problems, as it gets into the general population, that it does something else, nasty downstream. We just haven't looked at it close enough. People are aware that's one of the risks, of fast-tracking. A number of states have legislation on which people are considering, which will allow patients to get access to unproven substances, things still in trial. And the major argument has been, like I said to you before, the chance of something that's in a phase 1 actually working is small. Therefore, the chance of the toxicity and side effect is high. So while the FDA is very aware of this, and things are definitely moving through much faster than they were, which also saves not only time and maybe lives, but cost, et cetera, people are also aware the risk, is on the other side also, that we may fast track, we probably will fast track something into population that people are going to go suddenly go, whoa, we didn't understand that if you pass the, this particular immune drug, that 3 years later, everybody that got it, got some other new disease we weren't expecting. But that's the risk. But on the other hand, the benefit is obvious, and people are very aware of this area. Other questions?
My question is about eligibility for a trial.
Yep.
If I find out that a major institution is doing some research and conducting a trial in an area that I'm interested in, do I need to be a patient at that institution in order to be eligible to participate in the trial?
Somehow I missed saying this. It's a great question. The question, just to repeat it, is that if you're on a research study, do you have to actually be a patient at that particular institution that's doing it? So for example, if tomorrow, you find there's a study being done at the University of Pennsylvania, do you have to move your care there? And the answer is, like the rest of medicine, it depends. So, so if you're doing, if the study includes something which is a modification of what is generally being done in the community in general, it can often be done in your community and very much may be available directly in your community. If it involves a phase 1 study or something very new, you have to get care at that institution. Now, what we frequently do is we refer someone to a phase 1, that we're not doing, is that we'll continue to monitor the patient, work with wherever they're getting the therapy. So the answer is that people will get care on both sides. But you'll have to get a significant amount of your care and be monitored closely at the institution being, in the phase 1. You can't, if it's an experimental drug, it can't be done outside the experimental institution. And if it's experimental structure, that structure's got to be monitored in the institution. So a significant amount of your care will have to be given there, but you can often share your care with the local community. You shouldn't lose your local care. You should just be able to maintain those connections. It's like patients say, the worst thing you can learn by getting a second opinion, having a second opinion is you can learn something. And the same thing here. The best thing, worst thing that happens is you get other eyes, other people, to help to share with you and work with.
Yeah. Right now, what's the most cutting edge drug for stomach cancer?
For stomach?
Yeah, is there any like PD-L1?
Yes, there are some immunotherapy. It's actually Karuta, et cetera. Yeah, absolutely. I mean, the whole immune therapy is exploding, and absolutely for your gastric cancer. Again, it's not. I believe it's actually there's some early adjuvant work being done on study, and adjuvant work being therapy given after surgery at high risk, et cetera. But there's immunotherapy being done in the area. So the question is, if you look at the drug in front of you, it ends with MAB. MAB. So that's a code word for monoclonal antibody. That's a code. That means that the structure is so a monoclonal. So our bodies make antibodies through our entire lives to fight the cold, the flu, chickenpox, et cetera. That's what our bodies make antibodies for. And it was found some years ago in the early 80s that it was possible to design and make an antibody for a particular target. We can artificially make antibodies. And when you artificially make an antibody, it's all identical. It's the same exact thing. They all look the same under the microscope, et cetera. And those are known, therefore it's mono. They all look the same. Clonal, it's cloned, means manufactured antibody. So when you see that word, it means that substance, is a monoclonal antibody. That's how that works. It goes, in this case, a checkpoint inhibitor goes to the checkpoint, and the antibody attaches to it and shuts it down.
Because I noticed that it was all different kinds of
Correct.
medicines also, but I thought there was something.
Right. When you see those, so rituximab for lymphoma ends in MAB. That's a widely used clinical monoclonal antibody. Probably the oldest clinically used monoclonal antibody. Yeah, that's what that terminology means. It means something. Yep.
What's the role of NIH in Washington in the management of the clinical trials?
So they watch everything. So they set the standards, they set the goals, they set the broad picture. They fund $20 billion in cancer research, which fortunately this year was finally increased. Hadn't been increased in years, and it was finally now increased. They monitor directly the large trials. That include multiple different centers. They set up the standards. They frequently approve the therapies, et cetera. They're the greatest director, the greatest guide of all the studies that is happening.
Do all
They all are. Do they actually have to be? I'm not sure. Wouldn't surprise me, and et cetera, but there's no, given the fact that everybody wants to communicate.
So if I want to find a trial that's specifically targeted towards my type of cancer, I can go to that database.
Absolutely.
And I look it up
Absolutely.
by the type of
Absolutely.
cancer.
Right. You can. So if you break it, so it breaks down into pretty specifically stage, sometimes subtypes, subpathology, type of cancer. You know, what previously treated, not previously treated, multiple previously treated, you know, as well as geography, who's giving the study. It breaks down incredibly far. And then actually, you can then go and you can see the study. I mean, it'll print the details of the study and entering the study, and it's all right there. I mean, it goes on. And you can see that in patient or in doctor speak.
And last question. Can I find out which institutions are doing the most research in my type of cancer?
From there, you can. Can you search that study in that database in that way? The way I usually do it, if I want to get, is I change the zip code, you know, so to target that. But I'm not sure that it'll help you break it down that way. But what happens is when you bring up with the zip code, is you see all the studies and you can see the centers. It's all right there. It lists all the centers. It lists all the investigators, you know, for every study. And then, by the way, there's also phone numbers there's also phone numbers to contact at the centers, and just call people up and say, listen, I see you're doing a study. Is this study open? Usually, if it's listed, it is open, meaning it's taking new patients in, et cetera, and what are the requirements, you know, and you know, what's going on.
Is there any findings on the possible correlations between the HER2 with PD-L1? Like, if one is more expressed, the other one might be also more expressed?
I am not aware of any correlation between those two. It doesn't mean it doesn't exist, but I'm not aware of it.
So if you want to try the PD-L1 inhibitor drug, do you have to be tested positive for that expression?
No, it's PD-L1. For some studies, you do.
Okay.
See, you do.
You do that? That you have to be tested positive?
Et cetera. For some studies, you would. There's some evidence that the PD-1s work even in people that are PD-1 negative, so not all studies.
Is there any finding, like if once you get treated with PD-L1, but then afterwards the cancer come back and the cancer can mutate to a state where it actually beats the PD-L1?
I'm not aware of anything published on that. I don't know. Other questions? Well, thank you very much for coming today. You want to ask one more? Shoot.
One more. So, correct me if I'm wrong, but from what I understand after today's presentation, participation in a trial is not necessarily the last resort when other standard treatments haven't worked.
Yes. Participating in a trial may be the very first thing in some situations. It's you know, it's possible to be participated in multiple different trials over time, but not at all. Again, thank you very much for coming today. We really appreciate it.